Hi, welcome back to Animated Biology of the Cell. If you didn't see the previous part, I'd recommend watching that. In this video, we are going to take a closer look at DNA structure, strip it down to a basic model, and see how the properties amplify DNA replication accuracy. After we have delved into DNA details, we can discuss all the fail-safes in place that make sure DNA mutations are kept at bay. Without further ado, here it is in the most pure form I could find, a genuine DNA structure straight from a crystallography study. Look at its splendor, look at all the curves and the balls, and look at how from this angle it is incomprehensible. This is why I simplify my models, people. It saves you from details that don't matter. So let's simplify this model as well. First, instead of the space-filling model, let's switch over to a cartoon model with some schematics and explore some features. In essence, DNA structure is two backbones with a row of bases on either backbone, and those bases are also connected to each other. In general, this base binds with this base, and this base binds with this base. Adenine, thymine, cytosine, guanine. Now, just because these combinations are the most occurrent, doesn't mean there aren't any other combinations. It's just that those combinations are undesired and need to be fixed or prevented. But we'll get into that later. Let's just take a look at the backbone for now. If we number this structure starting from the base, we can see that the numbers 3 and 5 connect to these interspersed atom groups called phosphates. You may also notice that this has no symmetry from top to bottom, so we can define a direction. Here, if we go from top to bottom, it is 5 to 3. On the other strand, it is 3 to 5. This is what people talk about when they say 5' to 3'. It might seem like a detail that doesn't matter, but there are actually a lot of systems in place that can only travel in one direction and not the other. With that knowledge in place, let's talk about the meat of this video. How is it that replication as a whole only makes one mistake every billion bases? I mean, imagine it yourself. Your boss comes in with a billion pieces of paper, and you have to sort them all into 4 boxes. You know that you're going to make mistakes. Some mistakes won't get noticed at all, and some might lead to the death of the company. This is the task your DNA replication system has to perform. And it involves a 3-step process. We are starting with nucleotide interactions, then polymerase does some proofreading, and finally we are going to look at some new proteins that do a final check. So, nucleotides. Adenine really likes thymine and cytosine really likes guanine. Let's discuss why. If we take a look at their atom structure, there are 2 requirements before we can call this a correct match. A, the length of the base pair has to be this long, and B, enough hydrogen bonds have to be able to form. Adenine-thymine here is an example of good interaction. If we were to pair adenine with guanine or adenine itself, the resulting pair would be too wide to fit into a DNA strand. Adenine paired with cytosine, however, would have correct width, but wouldn't create the necessary hydrogen bonds. In adenine-thymine, there is a hydrogen bond from this hydrogen atom to this oxygen atom, and a hydrogen bond from this nitrogen atom to this hydrogen atom. Switch thymine for cytosine, and these bonds can't form. Hydrogen can't make a hydrogen bond with another hydrogen atom. The same can be said for nitrogen. Any other faulty combination of these 4 bases would fall under one of these 2 reasons. This has a success rate of about 1 in 100,000. Not a billion, but we're getting there. To make it easier for me to explain the next few topics, I am also going to use these more simplified models that are recognizable from a distance. Here is what they look like in assembled form. Now back to explaining. Sometimes nucleotides like to get a little sneaky. Due to natural processes, any of these bases can spontaneously change conformation. In this example, cytosine switches where the hydrogen atom is, and as a result, it is only able to bind to adenine. These conformational changes don't last long, and soon these pairs realize that they were not meant for each other. This creates what's called a kink in the DNA, and because of this kink, polymerase can't go on. When polymerase realizes this, it rips out every base it just put on the strand until right after the pair that was never meant to be. This, in a complicated term, is called exonucleolytic proofreading. The last line of defense against DNA errors is strand-directed mismatch repair. This needs to happen when all previous mechanisms have failed, and this strand is now stuck with a mismatch. Now, let's think about this. You see an obvious kink on the strand and you want to repair it using the original template. But which one is the original template? You can't know that when both strands are nearly identical. To distinguish young from old, there are so-called nicks in DNA every so often. The way these nicks get there differs between prokaryotes and eukaryotes. In prokaryotes, every occurrence of a GATC sequence is methylated on the A. This means that a methyl group is attached to the backbone over here. Methylating every GATC sequence takes some time, and so new strands aren't methylated yet. When a mismatch in the strand is detected, a protein called MutH will be called in. It will then nick every unmethylated GATC sequence. In eukaryotes, this process has a lot more to do with replication. If we go back to the previous video and remind ourselves of how Okazaki fragments are dealt with, RNase H removes RNA between Okazaki fragments, polymerase then patches up the holes that RNase H produces, and ligase fills in the final nick that polymerase can't fill. So, before ligase has done that, we can find nicks on the strand. Okay, you say, but that happens only on the lagging strand. Okazaki fragments only occur on the lagging strand. That's right. So there must be a system for the leading strand as well to have RNA inside the DNA. And recent research has actually pointed out that polymerase does incorporate RNA fragments into the synthesis of the strand. And after a pass from RNase H, this is where we can find our nicks. So, now that we can distinguish between new and old strands, let's introduce some new proteins. DNA mismatch repair, as we call it, is driven by the MutS and MutL proteins. And yes, from here on out, protein names will only get more technical. MutS detects mismatches in DNA and sends MutL to look for nicks.
What MutL does is not completely understood, but once it has done what it needs to do, it calls over a protein called EXO1 that removes a whole chunk of DNA. This is a job for a polymerase to patch up, and then the nicks left by a polymerase get fixed with ligase. You may have noticed a theme here with regards to polymerase. It only synthesizes in one direction, 5' to 3'. In this segment, I want to explain why. Nucleotides that are not bound together as a DNA strand have two extra phosphate groups attached to them. When polymerase binds one of these nucleotides to the strand, the extra phosphates detach and separate to release the energy needed to bind the nucleotide to the strand. In 5' to 3' replication, incoming nucleotides will have their phosphate groups pointed towards the place where a bond will be forming. In 3' to 5' replication, it is the last nucleotide on the strand that still has phosphate groups to use for replication. In 5' to 3' replication, nucleotides need to supply the energy to make the strand longer. The strand needs to supply energy to become longer in 3' to 5' replication. Here is why this will become a problem. We already know that polymerase will erase some nucleotides to fix a mistake it made. The moment it does so in 3' to 5' replication, the last nucleotide on the strand will not have the extra phosphates to supply the energy for the new bond. This replication fork has just shut down. This issue does not exist in 5' to 3' replication. If one spent nucleotide gets ripped out, there will always be another with extra phosphates to take its place. Onto the summary. During and after DNA replication, there are 3 systems to make the process as accurate as possible. First, inherent affinity between bases. Due to hydrogen bonds and relative base sizes, bases need to be paired with their correct counterpart to fit onto DNA. This has an error rate of 1 in 10 to the power 4. Next, exonucleolytic proofreading, where DNA polymerase realizes it just made a mistake and then goes back to fix the mistake it made. This has an error rate of 1 in 100. Lastly, we have strand-directed mismatch repair, which is mostly done by proteins other than polymerase and can be seen as a final proofreading step before finalizing replication. This has an error rate of about 1 in 1,000, so all multiplied together brings us to an error rate of about 1 in a billion. Thank you for watching.